Sunday, and I hope you will enjoy talking about ecology. It might be a bit misleading, the title, because, you know, I mean, it was supposed to be, it was meant to be the hands-on experience on statistical methods for community ecology, but that was designed for the previous versions of this summer course. But due to this COVID pandemic and the new order of the world, now the hands-on experience seems to be a little off. So probably won't be very functional and helpful. So we have to switch them to more theoretical discussions and how, I mean, potential examples of how you can use these statistics for the questions regarding community ecology. So we will be, for these 3 hours, we will be discussing some of the major concepts in ecology, and namely these will be dealing mostly with macroecology, and of course centered around community ecology, but we will be specifically dealing with species-environment relationships and species interactions. And I chose this picture specifically because that's a picture that I took over Greenland while flying to a field site. And you can see the patterns in nature are extremely complex. Here we are seeing aquatic ecosystems very near to each other. And you can easily see that some of the ones on the left top corner of the picture are quite blue. Some other on the bottom part of the picture are greenish, and the others on the top part of the picture is more pale blue. And all these colors are basically reflectance of light over these aquatic ecosystems. But this is a composite measure because light is produced, the reflectance of the light is produced by means of the abiotic components of these ecosystems plus the biotic component, the species living in them. So this is such a complex thing, how ecosystems are assembled and how they function. And what is driving them is a major question. Yes, and we will be dealing with some of the examples, how you can approach this complex topic using different methods or different perspectives. That's also important. Shall I continue with the lecture or shall we wait a little more? Elif or Efe?
It's up to you, Hocam.
It's up to me. Great. I suppose we have 40 participants in total, isn't it?
It seems 38.
In total. So how many we are missing?
I think Efe Hocam knows the best about the number of the participants.
I think around like 8 people. Yeah, so we are in a good number.
Okay, so that's a good number. All right. All right. Okay. The late ones can enter and then they can also watch on YouTube, so that's okay. So again, thank you very much, especially Efe and all the people who spent so much time to organize this. Also the supporters, TUBİTAK in this case. I should also show my appreciation. Plus all of you to be here on a Sunday noon. All right. So let me introduce myself a little. I'm Korhan Özkan. I am located at the Institute of Marine Sciences. I will show the location a little later. And if I need to provide you with my background, I was graduated from Middle East Technical University, Biology Department, for my undergrad and master's degree. For my master's, I was working or studying the experimental ecology, especially for the effect of nutrients on submerged macrophytes and the overall aquatic ecosystems, lake ecosystems. Then I had my PhD at Aarhus University, Denmark. It was mostly on community ecology of various organism groups. And since 2014, I'm a faculty at the Middle East Technical University Institute of Marine Sciences. So my research is mostly centered around community ecology and aquatic ecology, but my main methods that I employ for these studies is the experimental and field ecology. So I'm basically an empirical ecologist and spending a lot of time in the field. That is so much fun, but sometimes it is quite a bit of a burden or challenging, as you can see in the picture. And the current research areas as mostly, we can classify 3-4 topics, but we have been dealing lots of interesting questions in a variety of subjects. But we do quite a bit of work right now for the last few years on the ecology of high latitude and high altitude ecosystems. These are basically the polar high latitude and alpine high altitude ecosystems. And we mostly use paleoecology to understand the recent past of these ecosystems. We right now start a project on the aquatic ecology of arid landscapes and mostly the effect of salinization for these aquatic ecosystems. And as you can see, these are quite a bit extreme ecosystems to work on. And I will give some information about those too. We do work on land-sea interactions. We are located at the coast and we try to understand how the big ecosystems are interacting and eventually how it affects the communities along the coast. And as an ecologist, whenever you deal with the natural ecosystems, especially in this age, that's a bit pity, you are compelled to also work on conservation because we are losing the habitats, ecosystems, and the species very fast. And if you would like to know more about our research, you can check this ae.ims.meteor.tr, this is aquatic ecology.institute of marine sciences.meteor.tr. Yes. Oops. So that is our location. We are located at the Institute of Marine Sciences. That is on the northeastern corner of the Mediterranean Sea, facing Cyprus. But I'm not going to give too much details because I know that Mustafa and Arzu was here yesterday, so I suppose they already introduced our campus. Our campus is a fantastic place for research and living, but plus it's a good place for research, and I will give some examples later on. So just a few more details, what we are doing for these topics that we have been dealing recently. One of them is this high latitude ecosystems. These are basically polar areas. So we try to understand how the food webs of these extreme habitats, and namely here this Antarctic Peninsula, but we also work with Greenland. And we use paleoecology. Paleoecology means sediment cores, and you take some sediment cores and trying to understand what happened in the recent past. We also work with environmental DNA for more fast methods to understand the contemporary food web dynamics from scat samples of top predators. And we also use stabilized ops for understanding who eats who and where. But this involves lots of collaboration from various universities, from Ankara University, Istanbul Technical University, Koç University, Ulpan University, and Aarhus University. And so far, we have been to expedition to Antarctic Peninsula, and we are right now analyzing our samples. But we also, I've been to Greenland for 3 times, and we also continue our collaboration there. This is one part to understand the food web and ecosystem dynamics in polar regions. But the polar regions are extreme habitats, but they are not limited to that. These cold extreme habitats are also available in our latitudes. They are namely the alpine lakes in terms of aquatic habitats. And we also started a program to study these extreme environments. We have started expeditions to the alpine lakes of Taurus Mountains. These are the mountains just back of our campus, but they can go up more than 3,000 meters altitude. So that's quite extreme, and you can find permanent snow in the middle of the summer. And our main focus is again paleoecology. We try to take sediment cores and trying to understand how the ecosystems, these extreme ecosystems, have been shaped in the recent ecological time. You might be asking why that is interesting to use this extreme ecosystems. It is very good to work with the extreme ecosystems because they are simple and they are located at the extreme of the environment. So these are the first to respond to environmental change. And in general, the signal is clear because when you are working with more complex ecosystems, there are too many factors affecting, and it becomes very difficult to distinguish what's going on. then you need huge data, very long time effort, and so on and so forth. That's also a very, very important scientific endeavor. But for limited time and more clear signal, the extreme habitats are the best. So you can understand dynamics in a shorter time scale. And we also do it with lots of collaboration with Istanbul University and Untitled University and many others. And we got support from TUBİTAK and TÜBA for our work, and we are grateful for their support. Another work we do, that is the empirical part. We go to the field, we try to sample as much as ecosystems we have, and along a wider gradient of environment and biotic interactions, then trying to understand the patterns and the reasons. But this is in the realm of science, let's say the main understanding of science. These are pseudo or just halfway, because it's basically understanding the patterns. So we cannot do proper experiments. But that's also important for ecology to do experiments. So right now we are undertaking a huge project across Anatolia about this arid landscapes and the salinization of these aquatic ecosystems. And a part of this big endeavor, we are planning to establish 2 big mesocosm facilities in Turkey, one in Ankara, one in Mersin. And these mesocosm facilities are extremely nice way of dealing with proper quantitative science in ecology. What you do, you replicate the ecosystems to some extent in these tanks, and these are 4 tons of water in these tanks, including the sediment and everything. Then you can manipulate the environmental conditions or the biotic conditions, depending on your hypothesis, with controlled environments, with controlled replicates. And then you become much surer about the course of the dynamics and potential mechanisms behind them. And this was only possible. We are glad that Eric decided to move into Turkey. Eric is a leading scientist at freshwater ecology. He was also my PhD supervisor. And with a group of scientists in METÜ, from biology department, Meryem and Zuhal, we try to understand how salinization is affecting aquatic ecosystems across Anatolia and also globally, including expeditions in Kazakhstan that is planned for next year. If everything goes all right with the COVID, we will try to understand the dynamics in contemporary landscapes by sampling, then proper hypothesis testing in these experimental facilities. And lastly, as I told you, our campus is a great location for research because we are located at the crossing one big realm of ecosystems to others, land to sea. And they're interacting very, very intensely. That is, the most visible one is always the rivers. You know, the rivers are like drainages of veins of the land that's connecting everything on land to the sea, the coastal sea. But that is the visible part. The invisible part is the groundwater that is flowing beneath us. So, and it is mostly overlooked. You know, I mean, whenever there is this coastal eutrophication problem, eutrophication means that too much nutrients and the algal blooms and you don't want to swim. And in extreme cases, the fish start to die because of too much nutrients and the oxygen depletion. And dealing with the rivers is only a narrow part of this big interaction. So we established several monitoring wells in our campus, and we also got support from TÜBİTAK 1001 now to also understand in the entire Kilikien Basin, that is the water mass between Cyprus and Anatolia. Now we will try to understand how important the role of this submarine groundwater discharges for the overall nutrient and water transport to the sea. So this is what an empirical ecologist is dealing with, at least for my case, that involves the ecosystems, both for its biotic and abiotic components. And we try to use, as an empirical ecologist, we try to use both the fieldwork, expeditions, and the patterns in the space and time, plus proper hypothesis testing with well-constructed experiments. So what we are going to do for this theoretical part of this session. So I will try to give you a glimpse of examples from community ecology and macroecology, basically, and how you can use different approaches of statistics or different approaches of paradigms or a general understanding, conceptual understanding to understand these complex patterns. This will involve questions like changing communities, the patterns in space and time, what are the driving factors of these changes, the global patterns in diversity and the explanations for these patterns. Then we will go for conceptualizations like island biogeography. Then we seek some statistical approaches like null models. And I will give you an example, hands-on example from one of our research. Then we also talk about anthropogenic effects and biological invasions as novel factors affecting those things. That is the anthropogenic factors, our effects, both at the global and the local scale. There was a practical session for this original course, but for the COVID, we couldn't do it. Yeah. So what's ecology? I'm an ecologist, and the ecology is a science for sure, but that is also a phrase that is widely used in the society, because most of the times when we receive graduate applications, people mentioning that at least in our generation, I grew up with watching Costos or David Attenborough's documentaries, and I'm fascinated by the nature and I want to be a scientist. That is great. Nature is fascinating, but that is not exactly the ecology as a science. That's Attenborough with his fantastic Birds of Paradise. So I'm also a fan of Attenborough. I have all of his documentaries in my DVD collection, and I frequently watch them. Then you have the ecology for everything right now as a phrase. I mean, it is on the supermarkets, it is eco-friendly domestic products, it's ecological, organic food, and whatever. So lots of things as this etiquette of ecology. It has a sense of these complex interactions in nature and how we interact with the nature. However, again, this is not exactly the science part of the ecology. And we all know the Greenpeace fighting with the deep sea drillings or whale huntings. And that is the activism, and that's our reaction for all of the bad things going on with our nature globally. But again, this is not exactly the science, it's an ecology. So what kind of science when we mention ecology? First of all, ecology is the study of the relationships between living organisms, including humans, of course, and their physical environment. So how they interact. Most of the time it was, for the majority of time, it was dominated by observational studies and taxonomists. It was globally the case and still mostly the case in Turkey. But since the last few decades, it becomes an interdisciplinary subject. And with the availability of numerical methods and the empowering the biologists and ecologists with the knowledge of these numerical methods, it becomes more quantified science. Plus, with these experimental capabilities, it becomes even more quantified science. And it is important because science can be descriptive, which is extremely important to understand and catalog the life around us, because without this knowledge, we have no power to understand the dynamics. However, quantified science is also very important because, for example, a few examples, it becomes with less bias. You cannot exclude bias in science. We are human beings and we have our preconceptions. So definitely we will always have bias for one side or the other. However, when you do it quantified, then you can decrease the amount of bias in your conclusions. It allows you to understand complex dynamics. Human mind is great to recognize patterns, but what is driving this pattern is difficult to understand. And then you need high quantified methods, plus you need experiments to be able to understand complex dynamics. And it is also very important for improving our environment, because right now we are not living, most of the world is not living in a healthy environment. And to be able to adjust it in this phase of Anthropocene, the new geological era of human age, we need to have very precise ecosystem management. And for very precise ecosystem management, you need to know the thresholds very well, and you need to know how your ecosystems are responding. And this is only possible with quantified methods in ecology. So overall, quantitative ecology is the application of advanced mathematical and statistical tools and also experimental methods to any number of problems in the field of ecology. That's why it's also important to bring ecology from more descriptive to a more quantitative full science. Oops. Of course, this means this approach can act from local scale, like the experiments that you see a mesocosm in situ in the natural ecosystem mesocosm experiment, where you can test your hypothesis, to big data to understand global dynamics with less bias. So patterns in nature back again, that was the opening slide. And we see so many heterogeneity, both in the aquatic side of this environment and also terrestrial side of this environment. And the colors in the picture is one aspect of understanding it. And we see these patterns everywhere. One fantastic example of the patterns is the marsupials distributing across the globe. These orange areas are the places where you see marsupials. And I suppose majority of us are biologists, so we know marsupial is. These are the animals that have a pouch in their belly, so they nurse their prematurely born infant until they grow up. And what we see this pattern, it's intriguing. What we see is this orange colors are in part of the South America and the Middle America, and also Australia and the parts of the oceanic islands. So people would ask then naturally why? What happened? I mean, if they swim from South America to Australia, why they skip to Africa? So what is the potential reasons? Of course, the question will be in time because of the continental drift. Once upon a time, when the marsupials were diverged as speciation occurred, South America is connected to Australia through Antarctica. And that's why you know that it's about the biogeographic history that drives these patterns. So that is one approach to understand things. But when you keep looking on the globe, for example, here you see lots of colors across the world countries, and you see a big pattern. And this pattern is in the richness of bird species. So for example, South America is basically the hotspot of bird species. I mean, you can see 4,000 different birds, almost like 3,500 different birds in the neotropical region. But you go to, for example, Nearctic, that is the North America, only 700. So it is almost half of the world. And it's almost the same size. Why there are 4 times more bird species on the southern part, but not on the northern part of this almost same connected continent? The same thing is there. For example, Palearctic, that is the Eurasia, Europe and the Asia together, approximately 1,000 bird species. But when you check the sub-Saharan Africa, you got 2,000 species. Or the smaller Australia or Australasian realm, you see almost 1,500 species. Australasian realm is 1/6 of the Palearctic region. So why we see 1.5 times more bird species? That's very interesting. And the same patterns you see over and over wherever you look. For example, here you see the birds, the fish diversity. And there's a similar pattern, for example, that it makes sense maybe. There are 4 times more fish in South America than North America, and almost again 3 times more fish compared to the Palearctic. So almost similar with the birds, but not when you keep Australia. You remember Australian bird diversity was more than Palearctic, but when you check for fish, it is extremely low richness of fish in Australia compared to Palearctic. So what is the reason for all these changes? And these are the patterns in space. And in the meanwhile, maybe we should make an announcement about the progress. I would imagine if it would be much more helpful for everyone if it's an interactive session. So if you have any question or anything to say or add or discuss, we can do it anytime you like. Just open your microphone and ask it. Or otherwise, we will definitely have time during the breaks or whenever you like. But please don't hesitate to break whenever you need, you feel the need. So these are the patterns in space. But the patterns are not only in space. We see the patterns in time. For example, because of the great efforts of the ornithologists, both professional and amateur hobbyists, people count birds in a standardized fashion across Europe. And with this knowledge, we know the trends in birds, and that is dramatic. You see that the lines are decreasing, it's downward from 1980s to our date. We are losing the bird diversity across Europe. And you can see the arrows, and the arrows are big and red. So they represent dramatic loss. And the loss is everywhere. Oops. When you look at the forest birds, it's decreasing. When you look at all species, it's decreasing. But the most dramatic is 50% loss in farmland species. So again, you would ask why this is happening. And you see similar patterns. We can call it loss for majority of the organisms across globe, but let's call it change for now. And when you go to the diatoms, these are small unicellular planktons, plants in aquatic and part of the wet terrestrial ecosystem. And when you go to the circumpolar region, this is the area in the North Pole and all around the North Pole. Lots of paleoecologists were taking cores from the sediments and check for the change of the diatom species through the depth of the sediment, which represents through the time. Why? Maybe I should give some information about the paleoecology here. The lake sediments are great archives because they accumulate on top of each other. So the recent sediments, this year's sediments, all that material snowing through the water column accumulates as a layer on top of the previous layers. So if you can get an intact sediment core from lake bottoms or sea bottoms or from the wetland soil, you can have a chronology of things happened in this ecosystem. And some of the organisms are perfect to leave remains. And for example, diatoms, because they have this glass silicate shells and they're preserved in the sediment perfectly. And by checking them, you can see that some of the organisms are more or less stable through time. And some others are disappeared in the recent time. And some others have just been introduced in the recent time. So what is the reason? Climate change, industry, for example, nitrogen deposition from the atmosphere, and indeed these are the cases. So we see the change in time. And the patterns and complexities are not only in the polar regions or the Amazonian forests. For example, this is a landscape over Turkey. And I tend to ask this question whenever I show this slide. I suppose there are many Turkish students among us. The majority of the participants are Turkish. So I would like to ask, anybody knows what is at the center of this picture? So it's a silence. Oops. So this is a lake for sure. And of course, you don't know the resolution, but it looks like a big lake. And indeed, this is the biggest freshwater lake of Turkey. That's Lake Beşir, next to Lake Eğirdir. And we see a similar complexity in space across Anatolia. And of course, as an ecologist, we would like to understand what are drivers of patterns. But you might understand this, you might notice that this is the biggest lake of Turkey, freshwater lake of Turkey. And among our group, nobody recognized it. So these patterns, although when we look from space, is so clear, it was not always that obvious when you are walking over a space, because forests are huge and we can only walk only a small distance in relevant time. And it's all trees. So sometimes it's difficult to recognize these patterns. And actually, it was pretty late that the ecologists start to recognize there are patterns across the world. So otherwise, a person who only walked over the forests of Turkey, Bulgaria, or England would only see that forest. And if you don't see an Amazonian forest, it would be extremely difficult to understand the difference. And one of the very first recognition of these global patterns on organisms was by Merriam. We can call him a geographer. And he noticed that plants change through space, especially the forests. And it is not random. And he proposed that they are changing through temperature. And he even put, at this age, it was famous to put laws to understand things. Nowadays, we don't think like that. We don't think in terms of laws of nature. But he proposed that laws of temperature control of the geographic distributions of terrestrial animals and plants. And he even divided the latitude zones for boreal, austral, and tropical. And he said that he also put the zones in this region. But that was a great achievement. And it was only possible the world becomes united. People start to do trade in large extent. People travel and return written accounts of what they have seen. And this enabled researchers for global patterns, to recognize global patterns. One example is the evolution by Darwin. It was only possible he visited many locations across the globe. It was possible for Merriam to propose this law to be able to get accounts from various parts of the world of the animals and the plants. So it was recent. So that was the global patterns. But also it took us quite a while to understand the dynamics at the very local scale. And this is MacArthur, a very prominent ecologist from the mid-20th century. And he basically showed that to find big and relevant patterns in nature, you don't need to travel across continents. You might only need a notebook, a good amount of patience, and good observational skills. Because what he did, he basically struggled with the idea of amount of bird species in quite a bit homogeneous forest. So there is a pine tree, but there are 15 different species on that pine tree. So how they coexist? What he did, he sit down next to these trees all day long and then check which species is using which part of the tree. And he noticed that there is a pattern. Not all the bird species are using the same part of the tree. So they are basically resource partitioning in a way to coexist. And this is one of the remarkable examples of how you understand, how you resolve the patterns by using perspectives through species interactions. Of course, this one is a complex interaction in natural environment. But one of the pioneers of this was Georg Gauss. And he did one of the first observations of these species interactions at a controlled environment. That's also empirical science and hypothesis testing in controlled environment. What he did, he worked on paramecium species. These are unicellular organisms. And he tried to see how they grow on a good environment. For example, one paramecium species can survive and dwindle and make a population in a good growth environment. And he basically grows, and then at some point he stabilizes with the resources available in the environment. And another paramecium species also do that in isolation. So they can both grow in this growth medium. However, when you put those two together in this growth environment, one of them is taking over and the other is losing down. That was a big understanding of how species interact. And he phrased it as competitive exclusion principle. So you put two species in a controlled environment on a single resource. The good competitive one will win, and the less competitive one will lose. That was a big conceptualism. But you notice that according to this empirical evidence, there should be a single species. But how? That is the example how. The resource partition. So nature is complex. And when we consider the species, we tend to imagine individuals, don't we? Because species are basically individuals. I'm a human species, an individual from human species. So we always conceptualize interactions one by one. An individual interact with the other. But sometimes these interactions, biotic interactions, could be at larger scales. And one example comes from the shallow lakes. It was quite interesting for the aquatic ecologists, the limnologists. They keep visiting lakes, and they could see that at some certain conditions, you have similar environmental conditions, similar nutrients, similar temperature. But some of the lakes are extremely clear. Clear water, lots of macrophytes, lots of fish, diversity of fish, lots of zooplankton, and lots of birds. But when you go to other lakes, you have similar conditions environmentally, same nutrient levels, same temperature, but a pool of algae, a green pond with very less biodiversity, dominated by phytoplankton and almost no macrophytes. These are the submerged vegetation in the lake. And they noticed that later on, this vegetation is the key for this weird observation. And it depends on the nutrients and the feedback mechanisms coming from macrophytes. With this conceptualization, you understand that at low nutrient levels, you have clear water. Clear water will allow penetration of light to the sediment bottom, and you have macrophytes. At very high nutrient levels, it is all phytoplankton dominating the water column, shading out the macrophytes, so no macrophytes. But in the middle nutrient levels, it's a wide range. It can be both clear water or eutrophic algal dominated state. And it depends on the mechanisms of macrophyte. Why? Macrophytes can work as a refuge for zooplankton. Why zooplankton is important? Because they are eating up the algae, this phytoplankton. So the macrophytes, by providing a refuge for zooplankton, from the fish, of course, because fish will be eating zooplankton, and they provide a refuge for zooplankton, and zooplankton eats up the phytoplankton and provides light for the macrophytes. The macrophytes also stabilize the lake bottom, because most of the nutrients recycled from the sediment. And if you put macrophytes over it, they use the nutrients in the sediment, but the phytoplankton cannot, because the disturbance is not there, and the nutrients in the sediment is locked up, and they cannot reach the water column. Lots of complex processes. But basically, a group of organisms, which are the submerged macrophytes, can easily determine the entire ecosystem state. So there is also biotic interactions. An organism group is interacting with other organism groups, but this time it is more holistic. It's not only the understanding of individuals interacting, but the populations of individuals are interacting in wider temporal and spatial scales. Let me check time. So we should provide. Okay. Then I would suggest a break because it's only 2 minutes to quarter to 12. So let's have a break here and then have more patterns across the globe. Any questions or any suggestions about the progress? Are we happy or too fast, too slow? Any comments would be most welcome.